Hi, everyone, and welcome to our webinar. Today, you will hear about our SARS-CoV-2 reportavirus particles for neutralization. My name is Sharon Willis. I'm the Vice President of Sales and Operations at Integral Molecular, and I'm going to be the moderator today. So I want to start with some webinar housekeeping items. First, please remain on mute throughout the talk and use the chat, or there's a Q&A chat for questions. There will be a Q&A at the end of the seminar. And if we don't get through all of the questions, we'll follow up with email to answer any unanswered questions. At the end of the webinar, there'll be information on obtaining a free sample of the RVPs. A lot of you have already been through this process. It's a two-step process. You'll request a free sample and then be sent a link to fill out a Google Doc that'll capture your shipping information so that we can ship the RVPs to you, which we'll start shipping next week. So that's it. Oh, one more thing. We are recording the webinar, and everyone who registered will be sent a link. So now I'd like to introduce our speaker, Ben Durans, the President and CEO of Integral Molecular.
Great, thanks, Sharon. I'm glad to be able to talk to everyone today. Thanks for joining us. You know, at Integral Molecular, we've been working on viruses for the last 20 years, so we're proud to be able to support the research, the science, the vaccine development, the drug discovery that's going on for COVID-19. Our company in Philadelphia was founded 20 years ago by myself, Sharon Willis, Joe Rucker, a number of other people. It's founded on virus technologies. Is what we've been thinking about every day for the last 20 years, and mostly around envelope viruses and virus-like particles, things that we sell as lipoparticles. We've been making these for the last 20 years. And the last 10 years, we've been making reporter viruses, largely, well, not SARS-CoV-2, but other flaviviruses, retroviruses, and other types of viruses, flaviviruses, filoviruses, and we've been selling them to industry. So when SARS-CoV-2 came on board, we decided to turn our attention to that and offer these to researchers around the world as a reagent, as something they can use in-house, as something that can help in their own research and development of vaccines and drugs, hopefully accelerate the cures for everyone. Over the last 20 years, we've had over 50 manuscripts on viruses. The viruses are pretty wide-ranging, and we'll talk about some of them today. But again, making virus particles, making reporter viruses is something we've been doing for the last 20 years. And we're happy to be able to do it now for SARS-CoV-2 to help against the fight against this disease. Over the last 20 years, we've been growing, and we continue to grow. We work with over 300 different companies, sometimes behind the scenes. We're not always in the headlines, but we're very much behind the scenes, helping lots of different companies, many of which are recognizable. The ones that are publicly disclosed, we work with many others behind the scenes that are not publicly disclosed. And we've had over 250 patents and publications using our technologies, many in Cell, Science, Nature, other journals. We also continue to publish ourselves. Some of the journals on the manuscripts on the right, our own publications, working with collaborators, still working on viruses, working with virus technologies, antibodies, vaccines, and helping enable new therapeutics and new vaccines to get to market. So for COVID-19, we're able to offer a few different solutions, things that can help the research that's going on, the diagnostics, the vaccine discovery, the drug discovery, the antibody discovery that are going on. And all of these are available to anyone who's interested in using them. We work on both a fee-for-service basis and a collaborative basis. And actually, as we'll mention, many of these we can offer for free, collaboratively, because we want to make sure that everyone's moving forward in this fight against this disease. I'll talk mostly about the reporter virus particles today, which is, I think, why most people are here and probably what will be of most use to people. But we also offer a number of other things that are related to the RVPs that I'll mention throughout. One is called shock immunogenesis epitope mapping. It's a technology that we developed for understanding how antibodies bind to their targets. We've epitope mapped over 1,000 different antibodies with a 95% success rate. And over the next year or so, we'll expect to epitope map about 100 to 200 coronavirus antibodies. And that helps us understand the antigenicity of the spike protein, as well as protect intellectual property, as well as differentiate mechanisms of action. The other thing that we're doing on the COVID side is using our membrane proteome array technology. This is an array of 6,000 different membrane proteins expressed in live cells, so they're all natively folded. And we're using those for two purposes. One is for specificity testing against the entire array, mostly for safety testing of monoclonal antibodies. So before antibodies go into preclinical or clinical trials, they have to be tested for toxicology and safety. And off-target binding is one of the biggest risks for safety. So many people, aside from COVID and viruses, use our specificity testing. We're already starting to test COVID-19 antibodies for specificity. And we're also using it for some other purposes that I'll mention. So as I mentioned, over the last 10 years, we've been manufacturing reporter viruses, RVPs, reporter virus particles for R&D, for clinical trials. They've been used throughout the industry. We've been making lentivirus pseudotypes for the past 20 years and using them internally. And that's what our SARS-CoV-2 reporter viruses are based on, a lentivirus core, HIV core, and we've used that for Ebola, for Marburg, for chikungunya, for Hep C, as well as HIV, and now for SARS-CoV-2. And we've also made a different kind of reporter virus, we call the flavivirus reporter viruses. We use that for Zika, for dengue, and for West Nile virus. They're constructed a bit differently, but it's the same concept, where you have all of the structural proteins of the virus in their native antigenically intact form. But of course, they're easier and they're safer to use. They all encapsulate a reporter gene. They can use for infectivity, neutralization, enhancement. We also use them as biochemical tools for ELISA, biosensor, immunization, and phage display. And on the right are a couple publications that are relevant for this talk. The first one, the +1 paper, we published a number of years ago on dengue reporter viruses for all 4 serotypes. And we validated that technology and that those reporter viruses for clinical applications. And we recently put out a preprint, it's in review right now, for our Zika reporter viruses. And I'll refer to some of the data in those papers as we go along, because a lot of the things that we've done in the past, we're now applying to the SARS-CoV-2 reporter viruses for quality control, for manufacturing, and ensuring that we're delivering high quality reagents to everyone who needs them. As I said, we've been doing this for 10 years. So we've been able to work with a number of companies, many of which we're not able to name at this point, but some of which are public, like PaxVax, recently last year purchased by Emergent, and Takeda. We're both using our reporter viruses on Zika or dengue for many studies. In fact, our reporter viruses for dengue and Zika have been tested in thousands of patient samples at this point and feature in over 45 different publications, most of which come from other collaborators and partners, a few of which are our own as well. So on the SARS-CoV-2 reporter virus side, what we've designed is a reporter virus based on a lentivirus HIV pseudotype system. So we've taken the inside of the virus is the HIV core, and we've pseudotyped it with the SARS-CoV-2 spike protein, which is of course the envelope protein that's the spike protein that mediates fusion of the virus into cells. We've actually optimized the spike protein for high expression. We haven't changed the open reading frame or the antigenicity or anything important, but regions outside of the core protein we've changed in order to maximize the incorporation, the infectivity, and the expression of spike. But it's the native form of spike protein, just optimizing the system. So we're using this for ourselves and with partners and for customers for antibody neutralization, for serum screening, and any other kind of high throughput assay. And the nice thing, of course, about reporter viruses is that they're safe. They're designed with a 3 plasmid system to minimize any kind of recombination. These are not meant, I should say, for human use, of course. These are all for laboratory research use, so they're safe in a BSL-2 environment. They encode either a luciferase or a GFP fluorescent reporter. We have both available. And they're, most importantly, they're quality controlled for production. So I know many people are familiar with reporter viruses and pseudotypes. And the difference in what we do is that we do this on large scale. We've been doing this for 10 years, and we do it as a quality controlled reagent. Many of our reporter viruses have been through submissions in regulatory agencies, and we understand that they require more quality control, more validation as a critical reagent and validated assays in order to be used for clinical trials. So when it comes to larger scale trials, vaccine trials, reproducibility from lot to lot, from batch to batch, we understand those issues because we've been doing it for so long. So I'll show you some of the data. I'll make the large caveat that all of this is all being done in real time, and it's still very much ongoing, as is everyone's research. We are making and testing the reporter viruses and making them available as quickly as possible, but there's still many more experiments that are ongoing right now and that we're in the process of doing, but I'll show you what we can make available today. So we've made two types of reporter viruses. We've made GFP on the left and luciferase on the right. So for GFP, we've been able to get over 75% infectivity of target cells. And on the right, we've been able to get over 50 to 1, even more, signal to background with luciferase reporter viruses. So both are infectious, both are behaving the way we expect, and both are available. I should mention too that the cell type you use is pretty important. So as most people know, SARS-CoV-2 uses two different factors, receptors or other cellular factors to get into cells. So ACE2, the primary receptor, and the TMPRSS2 surface protease, which accelerates and helps the virus cleave and internalize and fuse. So these experiments are actually done with transiently transfected cells. And you can see in the orange cells with and in the blue without receptors. But the actual results, of course, are going to vary depending on what cell type you use. And I'll talk a little about that later. But we're making stable cells available with the RVPs as well. Again, no cost to you. If you want the cells, you can have them. We have a first version of the cells right now. We have a second version coming, but the first version is available currently. That will get very good infection. We've also been doing neutralization assays, and again, there's many more experiments that we're planning to do, but this is what I can show you today. We've tested these RVPs against patient serum. So on the left are two different patient sera that we obtained from recovered patients, and you can see both neutralize with the neutralize the RVPs, and a control serum in black has no effect on the reporter viruses. And on the right is a monoclonal antibody against SARS-2. We have many more coming, but this is the one I can show you today, where this antibody neutralizes actually pretty well the SARS-2 RVPs, whereas control antibodies in the open circle red has no effect. And actually in this experiment, we included another control as well, which is our VSV reporter viruses. So VSV is just a different envelope protein on the surface of the same fundamental reporter virus. And of course, it mediates entry of the virus in a different way. So the antibody against SARS-2 has no effect on VSV. The other thing I should mention, which is important, and I'll bring this up later, is in terms of the antigenicity of the virus, the reporter viruses, is that the control antibody we actually use for this purpose in the open circle red are actually is actually a control antibody against SARS-1. So that antibody actually binds the receptor binding domain of SARS-1, whereas this SARS-2 antibody that neutralizes binds the receptor binding domain of SARS-2. And that's important, of course, because when you start getting into epitope mapping and differentiating how antibodies are neutralizing and what the best correlate of protection is, these kind of differences matter. They both bind a highly important lesion of the virus, the receptor binding domain, but they have different effects. One neutralizes and one doesn't. And I should say this control antibody actually binds extremely well. It just doesn't neutralize. So there's important differences in epitopes that are important to recapitulate in reporter viruses so that you're getting the most accurate results. And that's important in some of our other studies that I'll show you. I'll mention this as well. The studies that we're doing right now for SARS-CoV-2, as I said, are ongoing. There's many more we're planning to do and are in process. We've done many of those studies before for dengue on the left and for Zika on the right. These are from two of those papers I mentioned, the 2011 Mattia paper, where we mapped, where we made and characterized reporter viruses for the serotypes of dengue, as well as the Whitbeck paper, where we've characterized the reporter viruses for Zika. Similar concepts in terms of making the reporter viruses and demonstrating their antigenicity, demonstrating reproducibility and stability, and I'll show some of that data. But I mention it here because these are the studies that we're planning to do for SARS-CoV-2, and when we're done with the SARS-CoV-2 experiments, a lot of the data for SARS-CoV-2 will look a lot like this set of data for dengue and Zika. The other thing I should say is that we've epitope mapped a lot of antibodies against dengue and Zika, over 200 antibodies, monoclonal antibodies against dengue, and currently over 100 monoclonal antibodies against Zika. We're doing similar things for COVID-2, but obviously that's just starting. But those epitope information gives you a lot of information about the reporter viruses, demonstrating that they're antigenically correct when you use them for neutralization assays, but also what kinds of antibodies, what kind of serum response you want to see, what's the most protective. And I'll show you some examples of that. We started doing some stability studies for SARS-CoV-2. These are the reporter viruses we use for infectivity, and the other slide. So again, these are preliminary data. We have many more coming, but we've done some freeze-thaw studies where you can see we've freeze-thawed it up to 3 times and still get essentially equivalent infectivity of the cells. You don't get infectivity of their non-permissive cells in white on the bottom. Interestingly, we haven't done a full curve yet, but we know if you leave them out put them in 4 degrees overnight, you do see a pretty significant reduction in infectivity shown on the right-hand graph. So again, we still have many more stability studies to do, but this gives you a preliminary sense of how you can use the reporter viruses and how you can't use them for the most benefit. On the bottom are similar studies we've done for Zika, doing time courses over 4 degrees, 25 degrees, 37 degrees. And of course, these are very important when you're doing neutralization assays because you want to know the timing of neutralization and how you're setting up the assays. We've also done freeze-thaw infectivity studies for Zika, as well as dengue. We've done all this for dengue as well. And stability studies over a week up to about 2 years to show that these reporter viruses are stable over years. So again, we'll be doing similar studies for SARS-CoV-2 and making all that data available to anyone who wants it, but it takes some time to do all those studies. We've also started reproducibility studies. These are really important in terms of as a critical reagent and for validated assays and for any kind of regulatory submissions. You want to make sure that when you use a reagent, it's not just a batch you whipped up on the spot and are using for that experiment, but it's something that's going to last you across all of your experiments to minimize bridging studies, to minimize variability, to satisfy regulatory, that you don't have to do more studies or bridging, that you're getting the same data from lot to lot, from patient to patient. So we started doing those experiments with SARS-CoV-2 on the left, getting similar results from experiment to experiment. But what we have to do next, which is shown on the right, is for Zika, what we did for Zika as well as dengue, is do different reproducibility, ideally using neutralization. So in this case, we did Zika reporter virus neutralization assays using an antibody where we could show we get the exact same neutralization, the same NT50 neutralization titer, 50% points, with different volumes of reporter viruses. That's important. So you show that you don't have any kind of defective viruses that are interfering with the experiment, that you are getting the same NT50s day to day. You can do the experiment on day 1 or day 2 or 5 or whatever, and you get the exact same data. And of course, from lot to lot, you get the exact same NT50s with lot 1, with lot 2, with lot 4, with lot 5, and so on. So you can minimize that variability. And again, that's not just variability of your assay, that's variability of your filings. That data ends up being reviewed, at least in regulatory cases, to make a decision about your drug or your vaccine. And so you want to have the most high quality data possible that goes into that data set. And the RVPs are something that can give you that when they're produced the right way. So this actually factors into a larger discussion, which is kind of summarized here. But what are the choices that I know everyone is trying to make right now for testing sera and monoclonal antibodies and other research projects? This is how the integral molecular reporter viruses stack up against other alternatives, at least a couple other alternatives that I know people are using. In terms of safety, speed, cost, capacity, the quality of the data, and whether it can be run in-house or not. The reporter viruses we offer are designed to make your life easy. They're safe, BSL-2, they're fast, you just add them, wait a couple days, 2-3 days, and you're done. They're relatively low cost. We can scale this up, as I'll show you, to liters for whatever capacity you need. Of course, you can use that in-house, so you have complete control over your experiments. And also, this is an important point, the data quality, as I said before, is really important. The data quality you get from a quality controlled reagent or reporter virus is very high. If you compare that with something like print, which is the standard that many people use, it's relatively low throughput, and we know it's BSL-3. No one wants to do those experiments in large scale in a BSL-3 hood, even if the capacity was there, which often it isn't. But even if you can do all that, prints also have this problem where they're actually relatively low quality. There's been many studies. There's one down here cited for Zika, but there's a similar study for dengue where they compared print assays across different laboratories, reporter viruses across different laboratories, and some other different systems for detecting infectivity. And invariably, print actually gives you pretty low quality data. It's actually usually pretty variable, and it differs a lot from lab to lab. So again, when you think about planning these experiments, you have to think about the power of the data that's going into your studies, and really comes down to the variability of the data that you're pulling out, especially if neutralization titer is an endpoint that you're making a decision on. If you have variable data, you're going to need more subjects and have poorer quality data. And if you have better quality, you're going to need fewer subjects and have better results. So it's something to consider as you choose different systems. Of course, I know many people are outsourcing this too. Not everyone has a BSL-3 hood or has done print. There's many good CROs out there. There's some variability as well. Most that I know of are using BSL-3 and print assays as the readout. Of course, you don't have control over those, and there's a lot of variability between different CROs. Many of them are good, but others I don't know. But those are decisions to make. And again, when you think about what you need for your system, there's many different factors. And I think the reporter viruses are a good alternative to many of the things that I see people discussing and deciding on. One other thing to note is that the reporter viruses that we're making are designed to be useful for studying vaccines. So serum response, largely against the spike protein, antibodies, monoclonal antibodies against spike, any other kind of spike drugs or peptides, or R&D that involves a spike protein. If your research or your vaccine in some way is not a spike target, the reporter viruses are not going to be useful to you. If you're going for some kind of, intracellular or non-structural protein of the virus, the reporter viruses are not going to give you the information you need. Similarly, if you're doing some kind of point of care diagnostic where you need the spike or the COVID-2 RNA in the virus, this is not the kind of system that's going to be useful for you. But for most people in the vaccine side and monoclonals and R&D, the reported viruses turn out to be extremely useful. So we've been doing this at multiple scales. We do this, again, we've been doing this for 10 years. So we've done large scale lots where we make liters and liters and liters of this, aliquoted according to customer specifications, do it under GLP quality and QC for regulatory purposes, minimize lots to minimize bridging studies. They've been tested in thousands of patients. So we're used to those kind of criteria and we know what's required. The flip side is that we also do this on small scale, and it actually turns out to be more important than you might expect, even for large scale studies. So, and I'll show you some examples of that. But for instance, when you're testing against COVID-2, of course, viruses evolve. COVID-2 is already evolving. And so invariably, there's going to be different strains. And of course, we already know there's MERS and SARS-1, as well as something like a VSV control. So there's different strain variants. Of course, when you're talking about antibodies or any kind of drug, there's escape mutations. And sometimes there's engineered variants of the protein as well that you want to test against. So in many cases, we've been able to make those very easily as well within our system. We can scale down the production of reporter viruses as well to make thousands of variants or a dozen variants, different strains or different naturally occurring mutations or different vaccine strains, whatever is needed in the course of testing either serum or monoclonal antibodies. And I'll give you some examples of that, of how it really ties into reporter viruses. So a few years ago, we published a PNAS paper on dengue viruses where we studied, in the end, about 3,000 different individual mutations that we made of dengue PRME, the envelope protein of dengue, across the different serotypes. This paper only covered dengue 3, but we did the same studies for dengue 1, 2, and 4 as well. And we've also done this for Zika now. Well, we've made alanine scans or other mutations across the entire envelope protein, partly for epitope mapping. We've epitope mapped over 200 dengue antibodies, but also for functional testing. So we already know which mutations within the envelope protein can knock out a specific antibody, can knock out infectivity, can knock out budding, can knock out receptor attachment in some cases. So we've already asked a lot of those questions, and that can get incorporated into vaccine design or escape mutation testing or other kinds of tests. In this case, we use it to build a model of how the envelope protein of dengue works, how it functions, which specific residues are required for fusion, for PRME latch functionality, for hinge functionality of the protein, and so on. We also use a similar approach to understand and engineer the HIV envelope protein, the GP140 trimer, where we used 850 mutations across the envelope protein to engineer the protein for better trimer antigenicity. And that was a paper we published a few years ago. As I said, we're doing similar things now across 1,200 different individual residues for the spike protein of COVID-2 for vaccine development. So all of that information goes into how the protein behaves, what parts of it are the most antigenic, either for protection or for lack of protection, many of the variable elements of the virus, for conformation, for yield, for safety, for stability. All those questions are things that we address as we make mutations, and we try to use that information in a smart way, publish it, and also we make it available to customers so that they can incorporate that information into their trials and their vaccines and their testing. The epitope mapping is worth mentioning. This is something that we do a lot of. We've epitope mapped over 1,000 different antibodies over the last 10 years or so. We have a 95% success rate, even for conformational antibodies, very dynamic envelope proteins that change conformation, that are oligomer specific, that are conformational, that are neutralizing, that are non-neutralizing. We've epitope mapped almost every kind of antibody, and we have over a 95% success rate. because of the way we do it. And there's more information on our website. I won't go into the details. But we've epitope mapped over 100 to 200 antibodies for Ebola, for dengue, for Zika, for Hep C, almost as much for chikungunya now. These are some of the results on the right for dengue, kind of summarized in one place. We've also done many other viruses and cellular proteins as well. But the key point I want to make is that these epitope maps become really important when you start talking about antibodies and vaccines. So they start differentiating mechanisms of action, how one antibody works versus another, or even how a vaccine is working. What kinds of antibodies are being elicited by a vaccine versus another vaccine. Many of our customers use this information for intellectual property. We've had many collaborations and customers use this information for designing cocktails of antibodies that bind in different locations, and even antibodies that may not compete, but or may compete but have non-overlapping epitopes. So they bind in very similar regions, but they actually bind very differently. For instance, the antibody that I showed you before that neutralizes SARS-CoV-2 versus another antibody that also binds in the receptor binding domain that binds perfectly well, but doesn't neutralize. So understanding the epitope and the very small differences between where these antibodies bind is critical when you're designing epitope cocktails of antibodies, as well as for vaccine engineering, to understand what specific residues of the protein are important for in inducing a protective immune response. So again, we're doing all of that in collaboration with many different labs. If you have antibodies against SARS-CoV-2 and you want them epitope mapped, reach out and talk to us. We do many of these things on a collaborative basis, i.e. it's free for you. Of course, we do some of these things on a fee-for-service basis too. Depends on what's being published and how much we can make available to the public. But if you're interested in epitope mapping, we do that every day. The other thing we do a lot of is a lot of specificity testing using another technology we call the membrane proteome array. I mention this because it is a little bit relevant to the reporter viruses as well in some of the studies that we're doing internally, but also for customers. This is an array where we put together all of the human membrane proteins, or nearly all of them, about 6,000 human membrane proteins, each one of which is expressed individually on live human cells. Because they're expressed on live human cells, it means we can do different things with them. We can do binding studies by flow cytometry, shown here on the top, and look for off-target binding of antibodies, for instance. Or we can use the same arrays in live cells to do infectivity assays by taking live virus, or actually the reporter virus in this case, putting it on cells and looking for receptors or attachment factors, or in the case of COVID-2, proteases that enhance the infectivity of the virus and allow it to get into the cell greater than the background. We've done this for dengue, and actually this is what's shown here is one of our screens against dengue. We've done this for the 4 serotypes. We've done this for Zika. We've done this for Ebola, Marburg, and several other types of proteins and cells, actually. But looking for new targets that may help us fight these different diseases. So we're doing that for SARS-CoV-2, putting reporter viruses on the membrane proteome array look for new receptors, if they exist, or attachment factors or proteases that enhance infectivity. And as I said, we've already started doing this for preclinical antibodies for specificity. That's one of the main uses for the membrane proteome array. And it turns out that specificity is the single largest predictor of safety in early clinical trials. So specificity testing for antibodies as well as for small molecules turns out to be really important. We only do antibodies for this, and proteins and biologics, not small molecules. But it turns out to be extremely important. And I will say that a surprising number of monoclonal antibodies, it turns out about 25%, are not specific to the target. They hit something else, and usually something very unrelated to what the target is. So, story for another day, but that's also available to anyone who's interested. So we're offering all of these things to anyone who's interested. The reporter viruses, the COVID-2 reporter viruses are available now. We'll start shipping them next week. It uses the Wuhan Q1 strain isolate. We're making additional isolates, and we expect that we'll have actually 1,200 different isolates or different mutations of them at one point or another in the near future that can be used for testing in any kind of way for research studies. We're also making COVID-1, MERS, and we're using VSV as a control, which often turns out to be very useful. And as I said, the target cells are also available. They're a little bit different for MERS versus the SARS, but we're making all of those as well. So for free RVPs, as I said, we're giving these away. If you're interested, there's only 2 steps you need to do. Click on our website, go to the contact page, and check this little box. You'll get an email from us, a form to fill out, and send that form back to us that has your address and your name and where you're from. Only qualified laboratories that have BSL-2 facilities, of course. We do require your billing information for shipping. So we do ask that you pay for shipping for these particles. We're not asking you to pay for handling or the viruses themselves, but just the shipping itself. But if you click the request and complete the form, send it back to us, we will ship out reporter viruses to you. Actually, we're not going to just ship out reporter viruses to you. We're going to send 4 tubes of the GFP reporter viruses to you. We'll send as well, included in this box, is a 1 tube of the VSV GFP reporter viruses. It actually goes a long way. They're also pretty infectious. Same kind of system. They're perfectly safe BSL-2 reagents, but very useful as controls to show that your serum or your monoclonals are not inhibiting VSV. Especially for serum, I should mention, serum can be pretty tricky, especially at high concentrations. You want to show that your serum is specific to the viruses that they're supposed to be neutralizing. We're also going to be sending you cells. So again, completely free. We'll send you these cells for infectivity assays. And I just take a step back for a second. The cells make a big difference. The cells we have right now are not fully optimized, but they're perfectly good for infectivity and give you high infection rates. If you want to use other cell types, you're certainly welcome to as well. Many people have reported that other cell types like Viros or A549s have been infectious, and they are infectable, but we found that they're not as not as robust as the cells that we have. So you can try any cell types you want, but we'll give you our stable cells so you can compare them if you like. But our stable cells in our hands give us the best results. We'll also include protocols. We know all of this is new, certainly for SARS-CoV-2. Many people have done virus infection assays or neutralization assays before. If you have your own protocols, that's fine. But we have starting point for protocols that we'll supply to you in terms of what we recommend, how much volume, and plates and things like that, media, incubation times. We'll also send you the safety data sheets, so you have all that on record, and technical information that goes with all of the viruses that we always ship. So as I say, if this is of something of use to you in your research, the reporter viruses, epitope mapping, the membrane proteome array specificity testing, we're here to help you. We're here to collaborate. We do all of this on a fee-for-service basis, on a partnership basis, in many cases, on a collaborative basis. And in this case, we're giving almost everything away for free. So we want to make sure that everyone is successful in their work on COVID-19. We all need to get back to our regular day jobs and win this war against the virus. So thank you very much. Thank you to Sharon for hosting, and if you fill out those forms, Layla, our account manager, may be contacting you to get everything set up for you. And thank you. I'm happy to take any questions.
Great, thanks, Ben. We have gotten a few questions, so we'll now enter our Q&A phase of the webinar. So the first question is, how many tests can be run from the free sample?
Yeah, so each tube is 1 mil aliquot, and from our tests, 1 mil is about right for 1 plate. So 196 well plate or 134 well plate, depending on how your assays are set up. We have some recommended volumes for each of the RVPs that get sent out in terms of what we would start out. Of course, we recommend you also do some dilution curves if you like to optimize things yourself, but each tube is basically designed for one plate.
Great. So I know that the free samples are coming with GFP reporter. For the luciferase reporter RVPs, what kind of luciferase is it, and what substrate do you recommend?
Yeah, so for GFP, it's an enhanced GFP. It's bright, and you run it by flow cytometry and microscopy. For the luciferase, of course, there's different kinds of luciferase. We went with Ranella luciferase for a number of different reasons. It's very sensitive. It gives very high signal to background, and if you use one of the glow reagents, this is very important. This is in our technical specs as well. If you use one of the glow reagents, the signal lasts a long time. You get much more reproducible signals from Ranella luciferase using the glow reagents that are commercially available. We don't sell the luciferase reagent, but we can make recommendations as to where to get it.
Great. All right, so another question is, how specific are these? Are you able, or are you detecting cross-reactivity with other coronaviruses?
Yes, that's a good question. So, so SARS-2, of course, is very different than SARS-1, as it turns out, and of course, even more different from MERS and other coronaviruses. And I think what's important too is we all know that this virus is continuing to emerge and evolve, and who knows what SARS-3 looks like at this point. And of course, many of the vaccine studies that are being set up are trying to anticipate what the next virus is going to be so we can prevent it before it even starts. So each reporter virus that we design uses a very specific clone of the envelope protein, of the spike protein. And of course, we define that, in this case, the Wuhan isolate. Other variants of COVID-2 will be made for reporter viruses, either custom if collaborators want specific strains or specific variants in there, or we'll be making additional strains available as well as they emerge and as they become relevant for studies. But of course, SARS-2 is very different from SARS-1, and those are good controls. The VSV as well, as I was showing the neutralization assays, those monoclonal antibodies both hit the receptor binding domain. Everyone knows that many of the monoclonals that are going to be neutralizing are going to hit the receptor binding domain, the RBD. But the fact that both bind, but one highly neutralizes and one doesn't neutralize at all, tells you that there's major differences between the viruses and even between the epitopes of where those antibodies are binding, which again comes back to some of the other work that we do.
Great, thank you. Can you say a little more detail on the cells that are being sent with the free kit? So, what's the background of the cells, that type of information?
Yeah, so we've actually designed a number of cells. We've made a number of cells to date. So we've tried to optimize the cell type for infectivity to optimize people's assays using Viros, using A549s, and some other cell types. But the one that seems to work the best for us internally are HEC293 cells stably transfected with ACE2. So that's the one we'll be sending out. I should mention that the one that we're sending out right now is a polyclonal because the clonal version is not fully completed yet, but we want people to get started. So we'll be sending out the ACE2 stables. They're not clonal, but we'll have the clonal available in June. And we're also continuing to make some enhancements to the stables to, again, enhance the assays further for sensitivity and reproducibility. So those will be available later in June as well.
Great. Another question, can these be used for membrane fusion experiments?
So of course, so reporter viruses, of course, are mostly measuring membrane fusion. You're measuring the fusion, the activity of the spike protein interacting with the receptors and allowing the virus to get into the cell. So it's designed for membrane fusion from that perspective. You can't really use these for like a cell-cell fusion assay or to measure lipid mixing. Those experiments are, theoretically can be done, but they're much more complicated. These are designed for one round infection. They go in, they don't get out, they don't replicate, but it is fundamentally a fusion experiment that you're looking at. And so neutralizing titers in this case are looking for neutralization of the fusion event to prevent the virus from getting in. If you have a drug that's working in some other way, for instance, a protease inhibitor, a SARS-CoV2 protease inhibitor, this is not the correct system for you. But if you're looking for an antibody or a serum or a drug that inhibits spike fusion, then this is exactly the right system for you.
All right. Another question, can the GFP be read quantitatively by a fluorescent plate reader?
Yeah, so we internally measure GFP a number of different ways. Of course, GFP is great because you can actually visualize the cells very quickly by microscopy. You can read with a plate reader. We don't recommend it though. The cell fluorescence, any kind of reporter viruses or no reporter viruses, even if you transfected cells in a plate with GFP directly, and every cell expresses GFP, plate readers are not that sensitive for detection. So we recommend flow cytometry for GFP detection and quantification. But if you have a highly sensitive plate reader type, like a mirror ball or something like that, then yeah, you could read it.
All right. Another question, is the spike protein full length?
Yeah, it's a full length protein. We have made some modifications outside of the core protein. We have modified the C terminus a little bit, as well as the N terminus a little bit, but nothing in the antigenic regions of the protein. But it's designed to mimic the exact antigen, the exact protein that will be found on live virus. Of course, those are studies that we also want to do and we need to do to show that the reporter viruses mimic exactly the antigenicity of the live virus, and we do that by epitope mapping, by neutralization assays, all ongoing. But they're designed to be antigenically identical. They're produced in human cells, and every other time we've done this for flaviviruses, for filoviruses, for chikungunya, for HIV, for Hep C, using all of that, we've been able to reproduce the native antigenicity in reporter viruses.
Okay, great. All right, here's another one. You mentioned there's a difference of percent infectivity of the RVPs on different cell types, for example, the Vero cells. How much difference of infectivity can we anticipate with the stable cell line compared to the Veros?
Yeah, that's a good question. The Veros have not performed very well in our hands, so you can try them, but I mean, you'll get something above background, but not that much. The stables that we're sending, that we can send right now, the ACE2 stables, I believe give about 25% infectivity. The transiently transfected cells that I showed in the data can give a higher amount, and that's transiently transfected with ACE2 and TMPRSS2. So a little bit different. Those give higher infectivity levels, and we're making stables from those as well, but they're not complete yet. So you'll get a little bit lower infectivity with the stables we send right now, and then we can provide the full stables at a later date.
Great. So what is the time course of infection? I guess how long until you can see the result?
Yeah, you can read about 2 days after infection. We recommend 2 or 3 days, depending on the signal levels you like and the signal to background and Z prime that you might need for statistical purposes. So basically, if your cells are set up, they're plated, add the virus and the antibody if you like, whatever you're neutralizing. Wait, incubate it for 2 or 3 days and read.
All right. Are we also developing SARS-CoV-2 VLPs, lipoparticles?
Yeah, those are planned. if someone has need for them. They're a little bit different. Those are more biochemical reagents. So as I mentioned, we use envelope virus-like particles, which we call lipoparticles. We use those for immunization, for biosensor, for ELISA, for phase display, many biochemical, biophysical methods, but they're great. The nice thing about the virus-like particle form of the spike protein is that it's in its native trimer form, and it's in the native membrane. So we find that when you leave the protein in its native state, instead of trying to refold it or produce as a monomer or artificially make it a trimer, you get better results. You get better antibodies when you immunize. You get better affinities. You're reproducing the actual avidity effect of the native virus. And in some cases, you get results that you can't derive using the monomer. For instance, a number of years back, we did a study with a collaborator characterizing some antibodies, and some of those antibodies on biosensor, some of those antibodies only recognize the trimer. So there was no way to do, at the time, those assays with a monomer because you couldn't get the antibody to bind. You couldn't quantify the kinetics of the antibody binding to the monomer. But if you use the virus particles as a biochemical reagent on a biosensor, we could get very good kinetics and show that it was a very different binding reaction than the monomer binding.
All right. All right. And another question, do you think that either the polyclonal or monoclonal stable cell line, when available, can be used with the luciferase RVPs to set up neutralizing antibody screens in patient serum?
Can you say that one more time?
Yeah, so the question is, will the RVPs on the stable cell lines, the luciferase ones, will they be able to be used with patient sera that have antibodies against the COVID-2 spike protein?
Yeah, exactly. That's what they're designed for. Exactly. So, similar to the data I showed for neutralization, if you have patient sera that have native antibodies that were derived from a natural infection or a vaccine immunization, the RVPs can detect those neutralizing antibodies in the assay.
All right. Let's see. Does the reported virus spike protein have the same glycosylation modifications on the virus?
Yeah, that's a good question. Yeah, that's a good question. We haven't quantified that yet, but we believe it does. And the reason we believe it does, there have been some very good studies published comparing the secretion of the spike protein in cell lines to the glycosylation of native virions. And it looks like the glycosylation is actually very, very similar. So we expect the glycosylation on our reporter viruses to be very similar as well.
All right. Another question, are RVPs going to be available for purchase to conduct optimization studies and screens? And can you also go through a little bit on how to place an order?
Yeah, so the first step, if you go to our website and click contact us and request the free RVPs, fill out that form, we'll send it to you to get started. At the same time or afterwards, if you want a quote on a large batch of them or different variants of them, or the luciferase version of the reporter viruses, Layla will be in contact with you, our account manager, to get you a quote. Again, we've sold these for years. We can send you a liter, or we can send you a mil, whatever you need.
All right. Another question, is the spike protein trimeric on the surface of the RVPs?
Yeah, it's functional. So the spike protein naturally forms a trimer, and it's the fusogenic form, the active form of the spike protein. And many of the antibodies that we've characterized for other viruses are oligomer sensitive, so we can prove that the envelope protein is trimeric. Of course, those antibodies are just coming now for SARS-CoV-2, so we don't have them yet, but we fully expect them to be. And in fact, the spike protein has to be trimeric, otherwise it wouldn't be infectious.
All right, I think we have time for one more question. So can you just reiterate when the other RVPs will be available, the SARS-CoV-1 and the MERS?
Yeah, we expect those to be, they're in process right now, and we expect those to be ready towards the end of the month in May or beginning of the month in June. And the cells for those are, well, for COVID-1 will be pretty much the same. For MERS, may take a little bit longer, but we're hoping in June.
All right, wonderful. All right, well, everyone, thank you again for tuning into our webinar. If we didn't get to your question, we will definitely follow up by email. Again, and we'll send out a recording of this webinar. You can also always reach us at info@integralmolecular.com with additional questions and to request a quote for additional RVPs. As Ben said, we'll start shipping out the free samples next week. And we look forward to working with all of you in the fight against this virus.
Great. Thank you, everyone.